My name is Carol Lee, as Netta said, and thank you so much for inviting me. It's a great pleasure and a great honor to be here to speak with you. Feel free to ask questions and feel free to show your face because giving online talks, it's very strange to talk to a blackboard. So I don't mind seeing faces if you happen to be okay with sharing that. And I'm going to mainly be talking about recent research in my laboratory. We have a paper in press in Nature Communications, and I'll be talking about that as well as some other recent work that we've done in my laboratory. So one thing I first like to mention is that populations throughout the globe are encountering unprecedented levels and rates of environmental change. Ooh, this is not working. So one of the most dramatic changes that are being caused by climate change is drastically changing habitat salinities across the globe. So if you look at this map here, which is based on data from 58 years, you can see that salinity has already declined in many coastal and high latitude regions, and salinity is increasing in many equatorial areas. So for instance, climate change is causing changes in precipitation patterns and enormous volumes of ice melt in high latitude. So many habitats in high latitude environments are already experiencing quite dramatic salinity changes. For example, if you look at the Baltic Sea region in this map right here, data from this time range shows that salinity has already declined in regions here in blue due to increases in freshwater runoff from increases in river flow. In addition to those factors, many populations are experiencing dramatic changes in salinity. For instance, if you look at invasive species, freshwater habitats are dominated now by brackish water species from brackish water environments. For example, if you look at the Great Lakes of North America, it is dominated now by brackish water species from the Black and Caspian Sea region. For example, zebra mussels here, quagga mussels, Cercopages. And what's really interesting about this is if you look at the actual observed number of invasive species, there's an incredible enrichment of these species from brackish water environments relative to what you would expect based on propagule pressure and transport opportunity. So these habitats are dominated and heavily enriched by brackish water invaders. So this is a very curious and remarkable pattern, given that if you look in aquatic habitats, there are definite biogeographic barriers caused by salinity. There's actually a very steep biogeographic barrier that separates saltwater and freshwater species assemblages, with the salinity of about 5 PSU forming a big biogeographic break across which most species do not cross. And this crossing of this salinity is incredibly difficult given the differences in ionic concentrations of the media. If you look at saline habitats, marine and brackish water habitats, they tend to have a high concentration of ions that are necessary for physiological functioning. In contrast, freshwater habitats are extremely deficient in these essential ions. So the question then is, what accounts for the ability of some populations to invade novel habitats when most of them cannot? And increasingly, we're understanding that this question regards the question of evolvability of populations and the specific nature of their evolutionary pathways. And in terms of the nature of evolutionary pathways, a key question that we often ask is, to what extent are evolutionary pathways deterministic? And you probably well know that Stephen Jay Gould asked this question. Oh, where's my cursor? Oh, there it is. If we rewind the clock and replay the history of life on Earth many, many times, will we consistently see the same outcome that is the reality we experience today? And his response to this question is no. If any replay of the tape of life would lead evolution down radically different pathways. And so he argued that non-parallel evolution would be the norm. And in fact, when we look at empirical results, many of you are probably aware of the many elegant studies, single gene studies that have uncovered striking cases of parallel selection acting on the same locus. And there are some really nice studies from the three-spine stickleback as well as other studies. However, if we look at quantitative traits, polygenic traits, those studies of polygenic adaptation have actually revealed non-parallel evolution with selection favoring different alleles among replicate lines. And the argument is that genetic redundancy would lead to alternative evolutionary pathways where the same trait optimum would be achieved through selection acting on different beneficial alleles. And these are some nice graphs from a review by Barge et al. Showing different alleles under selection during independent different evolutionary events. So the theoretical prediction, or what we could consider the null hypothesis, is, so this is a polygenic shift simulation and parallelism is on this axis, number of loci is on this axis right here. And so the prediction is that as the number of loci that contribute to a trait increases, you would expect the degree of parallelism to go down. And the argument, and this would be because many of the loci would be functionally equivalent and redundant, allowing many alternative evolutionary pathways to occur. So the null expectation is that polygenic traits should display genome-wide patterns of non-parallel evolution. And I've referred to some really elegant studies a couple of slides ago, where these key studies on polygenic adaptation, which have examined temperature adaptation in Drosophila, have been consistent with this null expectation. One thing I should mention that's interesting about temperature adaptation is that temperature has global effects on physiology, affecting all biochemical reactions and all metabolic processes in ectotherms. Thus, you would expect temperature effects to be extremely polygenic at one extreme end. So then that begs the question, what about other variables in other systems? And what other factors might be affecting the levels of parallelism? So some of the questions that we're interested in is what might be the role of population genetic structure in affecting levels of parallelism, such as selection acting from balanced polymorphisms. How might genome architecture of a trait affect the levels of parallelism, such as the number of genes encoding a trait, the number of loci, linkage among the loci, gene-gene interactions, such as the effects of positive epistasis? And what if the loci are not interchangeable and not redundant? What if a set of alleles and loci are absolutely necessary for adaptation to occur? So to address these questions, we are using the charismatic microfauna, the copepod, 1.5 millimeter plankton species, Eurythema affinis complex, to address these questions. Like Drosophila, they have a generation time of about 20 days. They're great to work with in the lab. And this species happens to be ecologically incredibly important because it supports many really important fisheries throughout the world, including herring, anchovy, and salmon. So the native range of this copepod spans high latitude distributions, coastal waters, and estuaries in North America, Europe, and Asia, shown here in pink. But within the last few decades, all within the last 60 years or so, this copepod has invaded many freshwater habitats, reservoirs, and lakes, shown here in blue. And my cursor disappeared again. I can't find it. So if we look at the freshwater invading populations, oh, there it is, shown here in white. These freshwater invading populations have arisen multiple times independently from genetically distinct clades represented by different colors in different geographic regions throughout the world. So there have been independent invasions from the Atlantic clade, shown here in red, and independent invasions from the Gulf clade, shown here in green. And these provide replicated tests for adaptation. So if we zoom in and examine this invasion of the Great Lakes from the St. Lawrence estuary, this invasion occurred about 60 years ago. And so first, let's talk about the nature of the polygenic trait under selection. So I'm first going to talk about some physiology before I get into the population genomics. So the question then is, what is the pattern of physiological evolution during this invasion event that happened within the last 60 years? So to address this question, we performed a series of common garden experiments where we took the saltwater ancestral population and the freshwater invading population, and we first reared them at a common salinity to remove the effects of acclimation. Then we took egg clutches representing distinct genotypes and split them across salinities to determine the physiological reaction norms or physiological responses of each genotype to the range of salinities. So I don't have time to show you all the data, but just in brief, this is the mean reaction norm for the ancestral saline population, showing low survival under low salinity and high survival under high salinity. And the freshwater population, which is less than 60 years old, shows the opposite pattern of high survival under low salinity conditions and low survival under high salinity conditions. So the question then is, given this dramatic shift in physiological tolerance, what exactly is the particular trait that's undergoing natural selection? What is the nature of this physiological trait that allows freshwater adaptation to occur? So let's analyze this some more. So a great challenge for a charismatic little animal like this copepod under declining salinity is that as environmental salinity goes down all the way down, to zero, the organism needs to maintain elevated hemolymph or blood osmotic concentration relative to the environment. So if we look at the ancestral saltwater populations from the St. Lawrence estuary and from the Gulf of Mexico, you can see they show the same pattern that as environmental salinity goes down all the way down to zero, these saltwater populations need to maintain an elevated blood osmotic concentration or hemolymph osmolality relative to the environment, this very steep concentration gradient. So let me ask you, what do you think the freshwater invading population, the freshwater derived population would look like? Would its hemolymph osmolality be higher under freshwater or lower than their ancestor? What do you think? How many of you think higher? How many of you think lower?
I would think the same.
The same. That's interesting. That's an interesting answer. Well, the freshwater derived population shows significant increases, evolution of significantly higher hemolymph osmolality relative to their saltwater ancestor, creating a much steeper concentration gradient with the environment. So what this means is we see an evolution of increased body fluid regulation or increased homeostasis, or the evolution of increased constancy, where this line is getting flatter relative to the environment. So this would be incredibly expensive. Maintaining this higher hemolymph concentration under freshwater conditions would require the increase in ion uptake from the environment and or a decrease in ionic losses. So I can't emphasize how much work this would be. And hopefully I'll impress that upon you. So the question then is, how would ion uptake take place and how would this trait evolve? So the action is in the ionocytes. And so the trait is embedded within the ionocytes, where there are ion transporters embedded in the membrane, and they perform the work for ion uptake and ion excretion. So ion uptake from a freshwater environment. And they are inside osmoregulatory organs like gills, kidneys, Malpighian tubules. And it's likely that ionocyte evolution of the functional capacity of this cell would be majorly important for crossing biogeographic boundaries. Yet before our series of studies, how ion transport functions evolve with environmental change had not been known for any organism, but we have done a bunch of investigations. So in freshwater conditions, there are several different hypothesized models for ion uptake in freshwater environments. So I want to emphasize that taking ions up from an ion poor environment is incredibly difficult and costly. So the big challenge here is to take up ions against a very, very steep concentration from an ion poor environment. So in this model, V-type ATPase, the proton pump, would pump out protons against the concentration gradient. And then using this proton gradient, other ion transporters would then bring, there is something not working here. Other transporters would then bring, my pad is malfunctioning. That's interesting. So if you see that NHA, I'm sorry, guys. I don't know why it's doing that. Let me clean my pad here. Okay, should be dry. All right, let's see if this works. That's better. So then this sodium transporter would then use this gradient created by this proton pump and bring sodium into the cell, and then sodium could enter the hemolymph or blood through sodium potassium ATPase. In this alternative model, ammonia transporter would transport out ammonia, and ammonia would react with protons, creating ammonium ion, and this deficit of protons would drive the sodium-hydrogen exchanger to export out protons in exchange for sodium, and sodium can then come into the cell with sodium potassium ATPase. Now, these models aren't completely mutually exclusive, but these are generally contrasting models. So the question then is, which model applies here? And there's a reason that I'm mentioning all these ion transporters to you. There's going to be a test at the end of this talk. And in addition to that, I'm going to be talking about these genes over and over and over again. And so there's a reason I'm presenting these models. So the question then is, which model applies here? Which ion transporters are involved? And how do they evolve during saline to freshwater invasions? So to examine the loci that might be under selection during saline to freshwater invasions, and to test those hypotheses, we performed comparative genome-wide gene expression analysis, where we compared the transcriptional profiles and replicate between saline and freshwater lines using RNA-seq on Illumina platform. So we found evolutionary shifts in genome-wide gene expression between the saltwater and freshwater populations. And here you can see to the right is increased expression in the freshwater lines, and to the left is increased expression in the saline lines, and thousands of genes showed evolutionary shifts in gene expression between the populations. And what's interesting is ion transporter genes dominated as the largest functional category that showed evolutionary shifts in expression. And to summarize those results, if you look at all of these gene paralogs, these genes and paralogs, in general, the genes involved in model 1 showed evolutionary increases in expression in freshwater populations under freshwater conditions. And it suggests that these loci contributed to freshwater adaptation. Now, I do want to mention that all of these genes have lots of paralogs. And what's really interesting is the different ion paralogs show different evolutionary and acclimatory patterns. For example, if you look at NHA, the sodium hydrogen antiporter, one paralog shows this pattern here, but another paralog shows a very different pattern. So we have different paralogs behaving differently in terms of acclimation and adaptation. And we wanted to explore these differences much more in much more detail. So what we did is we performed in situ immunolocalization of the ion transporter paralogs in the osmoregulatory tissues. And what we find is, especially when we look at the legs, which are like the gills, they osmoregulate by flapping, by using their legs. We find that the different gene paralogs show very different patterns of in situ localization, like where they're expressed, and very big differences in the degree of expression in different osmoregulatory tissues. So that was really interesting. And there's my cursor. Sorry, my cursor keeps on disappearing. So if we look at the paralogs of the sodium hydrogen antiporter, paralog number 7 here is expressed in the legs where they osmoregulate, and it's expressed on the same membrane and in the same cells as V-type ATPase, the proton pump, suggesting that these two cooperate in the osmoregulatory tissues. But there is no expression of the other paralogs, such as NHA5, suggesting that this particular trait and property is unique to this particular paralog. So to summarize what I just said, the ion transporters are the largest functional category that is evolving in response to salinity change between the saline and freshwater populations. The different ion transporter gene paralogs show distinct evolutionary and acclimatory patterns and likely have different physiological functions. And our physiological functional studies are indicating that these gene paralogs do differ in their evolutionary patterns and likely in their function. And one thing I'd like to emphasize is a few thousand different ion transporter paralogs are expressed in different cell types, different tissues, and different organs at different times and under different conditions. So these ion transporter paralogs are likely not interchangeable, and likely they are not redundant. So the question then was, what if the loci are not interchangeable and not redundant? What if a set of alleles and loci are absolutely necessary for freshwater adaptation? What impact would that have on the levels of parallelism that we witness? So that brings us to the next question. Across these independent saltwater to freshwater invasion events that occurred within a few decades, do we observe parallel selection acting on the same loci and SNPs in these wild populations during these independent invasion events? So based on theoretical expectations, the null expectation is that polygenic traits should display genome-wide patterns of non-parallel evolution. So what do we find? So to address that question, we examined genome-wide signatures of selection across independent invasions in the Atlantic clade, shown here in red, and in the Gulf clade, shown here in green. And this is a recent paper. This is work by my postdoc, David Stern. And so what we did is to examine genome-wide signatures of selection. We performed a whole genome sequencing of 100 copepods per population for these populations shown here. And what we saw was parallel shifts in SNP frequencies across these independent invasion events, going from the saltwater populations shown here in dark color to the freshwater populations shown in lighter color. And the gray lines are individual SNP frequency shifts, and the diamonds are the mean SNP frequency for that particular population. And you can see these SNP frequencies going from salt to fresh in the Atlantic clade and in the Gulf clade in the same direction. And what was quite remarkable about these SNP frequency shifts is the incredible high degree of parallelism, where 7,000 SNPs, among the 7,000 SNPs that showed any signature of selection across any of the invasion, 42.5% of them, or about 3,000 SNPs, showed parallel selection acting on the same SNPs across these independent invasion events. And 350 of the SNPs showed both parallel signatures of selection and an association with salinity. And interestingly, many of these SNPs that showed signatures of parallel selection and association of salinity occurred at or near ion transporter genes. So the highest gene ontology category was ion transporters, again, like the gene expression studies, with many several paralogs of NHA showing up, sodium potassium ATPase, and carbonic anhydrase. And in fact, the highest density of SNPs under selection resided within this genomic region that contained 7 tandem paralogs of the sodium hydrogen antiporter, that sodium transporter. And the blue dots represent parallel selection acting on the same SNPs across all the independent invasions we observed. And the size of the dot represents the level of association with salinity. And what was really interesting about these results is the very same SNPs of the SNPs that were under parallel selection during independent invasions. A pretty high proportion of them also showed signatures of balancing selection in the native range population, measured by similar frequencies or intermediate frequencies. So in these 4 distinct native range populations from genetically distinct clades in the Gulf of Mexico and on the Atlantic coast, the same SNPs that were under balancing selection, quite a few of them, showed signatures of parallel selection acting on the same SNPs during independent saline to freshwater invasions. So this was pretty surprising. We were surprised.
Sorry, may I just interrupt? What was the evidence for balancing selection that you have? That they occurred at similar frequencies, or what was your evidence for balancing selection?
Yeah, we used several different tests. We used similar frequencies and also intermediate frequencies. So we used a couple of different tests.
But for some of the examples that you showed previously for the parallel evolution, I saw the frequencies changing. What do you define similar? They started from close to 0% to whatever, I think the intermediate frequency in the different ones where you showed the parallel with the errors, yeah, this previous slide. So what is intermediate and where do you draw the boundaries? So that's, sorry for saying that, is because I find it super difficult to prove balancing selection. That's,
Yeah, I know what you mean. Why don't we, I can show you some graphs at the end of my talk. I have a bunch of graphs on this and we have a bunch of distributions and tests. So why don't I get back to that at the end of my talk? Is that okay with you?
Sure.
Yeah, it's in the supplementary file. There's a graph from the supplementary file for this, and it's easier to show you with a picture than me arm waving. Yeah, great question though. Thank you, Christian. So these ancient polymorphisms in the native range could provide genetic substrate on which selection can act during repeated invasions, promoting parallel selection during invasions. So these alleles that are under selection are at relatively elevated frequency. Okay, so it's possible that balancing selection in the native range and selection on intermediate frequency alleles could potentially promote parallel adaptation. So Christian, what we mean is intermediate frequency alleles that are at higher frequency than expected. But I could show you a graph on that. Thanks for the question. So given that we see these signatures of parallel selection in the wild populations, do we observe parallel selection acting on the same alleles in replicate lines during laboratory evolution experiments in response to salinity alone? So in the laboratory, we could more rigorously examine the effects of factors that could affect parallelism. And again, the null theoretical expectation is that polygenic traits should display genome-wide patterns of non-parallel evolution. So to address this question, so to perform these experiments, we took a saltwater population from Kiel, which is at the higher end of the extreme of salinity in the Baltic Sea. We took the saline population and we then split them into multiple selection lines, 10 selection lines, and we decreased salinity to 0 PSU, and we performed this experiment across 10 to 20 generations. And you can see here, this is the result for one of the 4 chromosomes of this copepod. It has 4 chromosomes. And we see that the SNPs that are under selection are distributed broadly across the genome. And these are different haplogroups shown by different colors. And in response to selection, going from saline to freshwater conditions, we see a rapid rise in beneficial alleles in the 10 selection lines, but no significant change in allele frequency in the control lines. And we measured parallelism using several different measures. This is work by Nathan Anderson, who should be in the audience today. He can answer some of the details on the simulations if you want. But we see here that our empirical data shown by these dotted lines are right here on these graphs. And we compared the results, the degree of parallelism between our empirical results to simulations of our data using different models of epistasis, and also compared to the multiplicative model where the effects of alleles are independent. So if we look at more detail in these graphs, in our data, on average, we saw about an 80% overlap in selected alleles between the replicate lines at generation 10. And this high degree of parallelism was pretty surprising. And the extent of parallelism in our data was matched only by the positive epistasis simulations shown here in yellow, and not by the multiplicative model, where all the alleles have independent effects, showed much lower levels of parallelism. So basically, positive synergistic epistasis, where the effect of an allele is increased by the presence of other alleles, was the only model that could account for the greater than expected parallelism that we saw, much greater than expected relative to the standard multiplicative model, where the effect of each allele is independent. And this is our empirical data, our results, level of parallelism at generation 10, and the level of parallelism at generation 6. So Nathan Anderson is working on this problem to much greater depth, and he's examining other signals of synergistic epistasis other than parallelism. He's looking at multiple measures to extract signatures of epistasis from our genomic data. And he'll be talking about that simulation modeling work at the ESEP conference in Vienna. So please go to his poster. He doesn't have a poster number yet, but I encourage you to seek him out and talk to him about all the work that we're continuing to do on this topic. So in addition to levels of parallelism, a high proportion of the selected alleles exhibited significant frequency shifts in all 8 of our surviving replicate lines at the end of our experiment. So there's a high proportion of selected alleles in all of the surviving lines. And only positive epistasis, so our data here is shown in brown, and only positive epistasis simulations showed a result that was similar to our empirical results in brown. And if you look at the null model of multiplicative, where all the alleles have independent effects, the proportion of selected alleles was relatively low. So as I mentioned to you earlier in the talk, the theoretical expectation is that as the number of loci encoding a trait increases, you would expect levels of parallelism to go down among the replicate selection lines. But when you look at our results, the level of parallelism was much higher than expected. And in fact, it behaved like only 20 loci that are under selection, suggesting positive epistasis. And so you would expect that functionally linked alleles would behave like a smaller number of alleles that are under selection. So we examined several other potential factors that could affect the degree of parallelism. We looked at the effects of physical linkage, and we found that the effects of physical linkage was quite minor. We also looked at the effect of balanced polymorphisms, or the effect of starting allele frequency, and we found that its effect was relatively low and could not explain the high degree of parallelism that we found, but it did seem to facilitate in the positive epistasis simulations. So while the effect of starting frequency was relatively minor, it did seem to have a facilitating effect on the positive epistasis scenario. So these factors alone not...
Sorry for interrupting again. Did you find increased levels of linkage disequilibrium between pairs of the genes that you think show positive epistasis?
No, not at all. Good question, though. That's a really good question. We did not. So these factors alone could not explain the high degree of parallelism that we found in our data. So again, if we look at the loci under selection, like our previous experiments on the wild populations from North America and in our gene expression analyses, again, ion transporter paralogs dominated as the functional category that showed signatures of selection in our laboratory experiment in our replicate lines. And again, here you see paralogs of carbonic anhydrase and paralogs of the sodium hydrogen antiporter. And I'm not going to point because it seems to advance my slides when I try to use the cursor. And interestingly, the SNPs under selection in the laboratory were also showing signatures of selection in the wild Baltic Sea populations. So if you look at the background loci, the null genomic distribution of loci and their QX score, which I'll call a signature of selection here, the loci that were under selection in the experiment that were present in the wild showed an extreme signature of selection relative to the background loci in these Baltic Sea populations. So these are the experiment selected SNPs, the SNPs that showed signatures of selection in laboratory selection. They were also in the wild populations. And if you look at the distribution of, I would call them signatures of selection scores, the experimental selected SNPs were outliers and showed signatures of selection. Now, the other thing that was really interesting about our results is that the SNPs that were under selection in our laboratory selection lines, the ones that increased in frequency with declining salinity, these laboratory selected SNPs were at higher frequency and were found in greater abundance in the freshwater regions of the Baltic Sea. So that says, that indicates that there was ecological relevance for the SNPs that were under selection in response to low salinity in the laboratory, as they were also showing signatures of selection in the wild. So it does appear that genome architecture appears to be important in terms of the degree of parallelism we saw. We saw an extremely high degree of parallelism in our laboratory selection lines, and it appears that positive synergistic epistasis might be playing a really important role. And as I mentioned to you, we are continuing this study and looking for multiple signatures of epistasis using different genomic parameters in our continuing study. So the question is, do we see parallel selection? Yes, we see parallel selection acting on the same loci and same SNPs in the wild population, as well as in our laboratory selection lines, despite polygenic adaptation. And our results indicate that parallelism is likely driven by positive epistasis and might be facilitated by selection on more abundant alleles. On selection on standing genetic variation in the starting population. So this potential role of epistasis is consistent with mechanisms of ion uptake that I talked about earlier in my talk. And this requires the action of multiple cooperating ion transporters to accomplish the role of ion uptake from an incredibly ion-poor environment. This is a challenging feat that needs to be accomplished. So it makes sense that specific combinations of alleles for sets of cooperating ion transporter paralogs would be adapted to function together, and they would be favored by natural selection as a group in response to environmental change. And so to summarize what I've talked about, again, to emphasize the point, we see extremely high levels of parallel evolution acting on the same alleles, repeatedly favored by natural selection in wild populations and laboratory selection lines when we use very different populations from genetically distinct clades in different continents. And this parallelism is likely driven by epistasis, though we're exploring this question much further and in greater depth, where specific combinations of alleles across ion transporter paralog genes might be favored by selection together. And it's likely that balancing selection in the native range might be facilitating this phenomenon. So this study is novel in demonstrating that positive epistasis can promote parallelism on a polygenic genome-wide scale. And so I'd like to also mention that this mechanism observed here might be relevant for other systems where rapid adaptation to environmental change is occurring. As I mentioned to you earlier in my talk, many organisms are experiencing this dramatic salinity change and rapid habitat change in general due to climate change. And this particular mechanism that I talked about today might be more common than previously has been recognized. And with that, oops, again, my keyboard, nothing is working. Okay, and with that, I'd like to acknowledge the members of my lab whose work I talked about, including David Stern, postdoc, Nathan Anderson, a graduate student, Teresa Pop, Juanita Diaz, and others, as well as my collaborators at University of Montpellier, where I have a satellite lab, and my funding sources, including from the National Science Foundation and the French ANR. And with that, I'd like to conclude by mentioning that we are looking for a postdoc to do theoretical work, to take our empirical data and apply it to evolutionary rescue models. And our collaborator on this grant is Luis Miguel Chavan, who is right there with Christian Schlatter. You're probably chatting daily with him. And so if you are interested in this postdoc position, which is a beautiful integration of empirical data with theory, please contact me. So thank you very much. I appreciate your attention. Christian, thank you for your thoughtful questions. Any other questions?